Today we're going to start our unit on the central nervous system and the brain. Now looking here, the central nervous system is going to be composed of the brain and the spinal cord. Then we'll discuss the peripheral nervous system. The peripheral nervous system is going to be composed of the spinal cord and the cranial nerves. So looking here, you'll find that with the central nervous system, it's often referred to as CNS. So if you see that in the notes, CNS is going to be your central nervous system. It's going to be made up of the brain and spinal cord. Coming down with the brain and spinal cord, we also have it divided into what's coming off of those, and that's going to be referred to as the peripheral nervous system, or the PNS. And so the brain and spinal cord are the first, the CNS, and then what comes off of those main parts is going to be the peripheral nervous system. And so that's going to consist of nervous tissue outside of that central nervous system and the ENS. So coming on down, what these things do, these are going to act as part of the afferent division. What happens is they are the receptors. They're going to be picking up on things. So you have these special sensory receptors. These are going to help monitor things like smell, taste, vision, balance, and hearing. And so they're going to pick up on these things and they're going to travel up with sensory information that needs to be processed by the brain. Also here, you're going to have the visceral sensory receptors. These are going to monitor your internal organs, so they will take in information and carry that to the brain as well. And then you have your somatic sensory receptors, and these are going to monitor skeletal muscle, joints, and skin surfaces. So they pick up that information, and they're also going to carry it up. Ooh, not sure what happened there. Hold on. There we go. And so it's going to pick that up and take that sensory information through the afferent pathway, and it has to go to the brain in order to be processed. So from there, that information processing is going to be integrated, processed, and coordinates that sensory input and decides what type of motor commands needs to come out. So what needs to be done with the information that's coming in, if anything. So what happens? The motor commands within the efferent division or efferent division are going to come out after the brain decides what to do as part of the CNS, and it could go through the somatic nervous system or the SNS, and that's going to lead to the skeletal muscle. So that will tell the skeletal muscle if it needs to contract or release, if it needs to get you from one place to the next, or it could be coming out through the autonomic nervous system or the ANS, and there is going to be divided into two parts. You'll have what's referred to as the parasympathetic division, and you'll have the sympathetic division. And so we're going to go over those in detail in a little bit. So with those, they're going to control things such as smooth muscle, cardiac muscle, glands for the parasympathetic. And with the sympathetic, it's going to be all of those and add in the adipose tissue. Looking at the brain here, it's going to be a very complex organ. So going over the first part, we have what makes up what we consider to be the main section of this organ. That's going to be the cerebrum. So in with that cerebrum, that's going to control major things such as conscious thought process, intellectual functions. It controls smelling and hearing, seeing, things like that. It's going to have memory storage and processing there, conscious and subconscious regulation of skeletal muscle contractions. Next, we have a section that's going to be found deep within the brain. You can see actually how it would be inside of the brain if the outer portion of the cerebrum wasn't covering it, and that's going to be referred to as the diencephalon. So the diencephalon is going to be made up of two parts. First, you're going to have the thalamus. The thalamus is going to be there to relay and process sensory information that's coming in. Next, you're going to have what's referred to as the hypothalamus. That's going to help control things such as emotions, autonomic functions, and hormone production. It also helps control things such as temperature inside of your body. Coming on down to the midbrain, that's going to be found right underneath that diencephalon section. And so that's going to be in green there. So the midbrain is responsible for processing visual and auditory data. It's there for generating reflexive somatic motor sensory responses, and it maintains consciousness. Next, we come down to the blue section, which is going to be found through here, and that's referred to as the pons. The pons helps relay sensory information to the cerebellum and the thalamus, and subconscious, somatic, and visual motor centers are going to be found there. Next, come over to below that pons section, and you'll have what's referred to as the medulla oblongata. This is going to relay sensory information to the thalamus and to other portions of the brain. The autonomic centers are there for regulation of visceral function, things such as cardiovascular, respiratory, and digestive system activities. And lastly, we have the back section of the brain, which we see here in the light blue. That's going to be the cerebellum. So don't get your cerebrum, which is the large section, and the cerebellum confused. Cerebellum is going to be a smaller section towards the back of the brain. And this is going to coordinate complex somatic motor patterns. And it's also going to help to adjust output of other somatic centers in the brain and spinal cord. Next, we're going to talk about the coverings of the brain. So your brain's not just going to be in your skull open, it's going to actually have coverings that go around it. And these coverings are referred to as the meninges. They're going to consist of 3 layers. You have what's referred to as the dura mater, the arachnoid mater, and the pia mater. They're going to be composed of a very tough connective tissue, and inside that connective tissue, you're going to find blood vessels and nerves. Now, with those nerves that are going to be there, they're actually going to be able to pick up on pain. So this is one part of the brain that does have feeling to it. So if they were to go in and do brain surgery, they're going to want to make sure that they used anesthesia in order to numb that part up so you don't feel it when they cut through it to get to the part of the brain or the surface of the brain that they need to work with. Now, once they go through the meninges, there are no sensory receptors that are going to be found within or on the brain that's going to pick up on pain. So you don't have to worry about anesthesia for the actual brain, but you do within the meninges. So going into the dura mater, that's going to be the outermost layer of the meninges. It's going to connect the meninges to the skull bone, so it forms a cover over the inner portion of those cranial bones. It's also going to cover and protect the spinal cord, so it not only covers the brain, but it runs the entire length of that spinal cord. The arachnoid mater is going to be the middle layer of the meninges, and it's going to be found between the arachnoid mater and the pia mater. You're going to have what's called a subarachnoid space. Now with that space, that's where we're actually going to find our cerebral spinal fluid, or sometimes you might see that within the notes as CSF. So that's going to be the spinal fluid that's going to be circulating around the brain and then through the spinal cord. So if they were to do a spinal tap, that's the fluid that they're going in to get, the CSF, or cerebral spinal fluid. And the innermost layer is going to be the pia mater. That's going to attach to the actual brain and spinal cord. It's going to be rich in blood vessels, and it's going to nourish that central nervous tissue that's going to be found there. So looking here at the picture, if you go in, you can see these meninges here. So you see that dura mater is going to be in the whitish gray part there. The arachnoid mater is going to be found there in the purple. Then you see that open space, that's the subarachnoid space that you see. It's pointing to that blue section. That's where you're going to find that cerebral spinal fluid, or CSF. And then on the outer portion, you find the pia mater. And so that's going to be found in red. And so that's going to cover the brain completely. And notice if you look over here at this picture, it not only goes around the entire brain, but it also will go down the spinal cord. So looking at it as you go down through the spinal cord, you'll see the spinal cord in the center. You'll see what's referred to as a ventral root that's coming out. That's coming off of that spinal cord. That's going to go into the peripheral nervous system and connect the could connect the arms, the sides, the legs, anything that's coming off that spinal cord. Those are going to be the nerves that are leading off of it, and they'll continue to get smaller and smaller as they go to the different sections of the body will that actually have sensory receptors that pick up on information. Now going in, you can see that the innermost layer is that red layer, and see how it wraps around that spinal cord. Then you'll see the purple layer, which is the arachnoid mater, and that's going to be wrapping kind of around that pia mater, but notice the blue in between. That blue in between is that CSF, that cerebral spinal fluid. And then wrapping around on the outermost layer is going to be kind of that clear light blue or gray layer. That is going to be the dura mater. And so if you look over at this picture, you can see how it looks if you were to do a cross section or transverse section and look down on it. Right? So in this picture, it's kind of showing you a different view. It's showing you a side view or a sagittal view that cuts in. And looking, you can see how these meninges will go around the main part of the cerebrum of the brain. And then if you look at this section a little bit closer, that's what you see up here. And so you'll see the cerebral cortex, part of the brain. Then you'll see the pia mater, which is going to be this lighter section. You have your subarachnoid space. That's where you're going to find your cerebral spinal fluid. And then you have your dura mater, which is going to be the outermost portion. Now here it also shows what's referred to as the dural sinus. Now this dural sinus is going to be an open cavity that's going to be able to carry fluid, that's going to allow, it's actually going to be part of the venous system, which will transport things from the blood into the brain and take things out. So here's another view. It's going to be another sagittal view or a cut that you can see. So you can notice your pia mater is going to be that innermost covering. So you see that in red there. Next, you'll see the subarachnoid space that is open and has that cerebral spinal fluid flowing through it. Notice how it flows up from one way and up from the next way. Notice how it can enter that dural space. That dural space does have your venous system or your blood flow flowing through it. And so this is what helps with that blood-brain barrier there. Also know the arachnoid mater on the outer portion, and then that dura mater. So looking at this picture, this is going to be a side profile picture. You're going to see the different ventricles. Now the different ventricles are going to be hollow chambered spaces. And so first we'll start off, you're going to have two lateral ventricles, one on each side of the head. It's going to be filled with cerebral spinal fluid or the CSF. You come down to the interventricular foramen. That's what's going to connect these two lateral ventricles together. And then further on down towards the center of the brain, you find what's referred to as the third ventricle. And then it comes down into what's known as the cerebral aqueduct. And that's going to carry the cerebral spinal fluid to the fourth ventricle. And from the fourth ventricle, it goes into what's known as a central canal. Now, all of these are going to have that cerebrospinal fluid running through them, and it's going to be in a very important fluid that's going to go throughout the center of the nervous system here. So this is a closer look inside of the brain, and here we're going to see more of that blood-brain barrier. So starting off, get my pen. First off, we have what's referred to as the choroid plexus, and the choroid plexus is going to be responsible for having these cells here, which are going to be called the choroid plexus ependymal cells. So these ependymal cells that are going to be found in this section here, from here on through here, are going to be the ones that are responsible for making the cerebral spinal fluid. And so when they make this fluid, they're going to excrete this fluid into this subarachnoid space, and they're going to have it there. It's going to help nourish and keep the brain and spinal cord healthy and fed. Now, with this, you'll see this section coming through here. This is actually going to be a capillary. So within this capillary, it's delivering nutrients, and it's also delivering things such as oxygen and things that the cells need. What's going to happen is it's going to diffuse across these ependymal cells and go into the cerebral spinal fluid that you're going to find here in the ventricles. Now, if there's any waste products from the brain or from anything inside, that is then going to diffuse into the cerebral spinal fluid. So here's going to be part of the brain. It's going to diffuse into the cerebral spinal fluid. Then it's going to diffuse into these choroid ependymal cells, and from there diffuses across into this capillary. Then this capillary will take those waste products to the kidneys, where the kidneys are then going to filter them out and clean the blood or filter the blood. So here, notice with these special ependymal cells, they're going to have this microvilli on the ends there that increases the surface area so it can absorb more waste products and things that it needs to in order to come across here. Now, with the things that are coming into the cerebral spinal fluid, they're going to flow across that membrane. So notice how the blood and the cerebral spinal fluid never actually touch. And that's going to be what's referred to as the blood-brain barrier. It's going to have to come across these ependymal cells in order to get into that fluid. And then the waste products from the brain or that are in the fluid are going to have to come across these cells to get into the capillaries. So they never actually mix. Now, with these cells that you see with the microvilli, these choroid plexus ependymal cells, they're going to be held together with tight junctions because we don't want things to be able to flow back and forth with each other. We only want certain things to diffuse across. When you come over to the regular ependymal cells that you see that are going to be going around the between the cerebral spinal fluid and the actual brain, which is up here, you're going to see that there is a lot of opening and a lot of space, and it's going to allow things to flow in and out to feed that brain there. So this picture here is going to show you different places that we find these choroid plexuses where they are making that cerebral spinal fluid and where they're allowing things to diffuse across. Now, they're not going to be found everywhere within the system, but this is going to show you some specific places where you will find these choroid plexuses. And so here you're simply seeing a section of that superior sagittal sinus. This is going to be where the cerebral spinal fluid is going to exit out from that arachnoid or subarachnoid space, and it's going to actually come into this superior sagittal sinus where it then could enter into the blood, because you don't want too much cerebral spinal fluid and you don't want too little. So if you happen to end up creating too much with those ependymal cells, it could actually be filtered out through the blood by going into the bloodstream from that point. But that's where it's going to exit out and be filtered from. So we talked about the different fluids and how they're made for the cerebral spinal fluid, but here's actually going to be a picture of the brain. And so with the brain, we have, remember, the big, large portion of the brain, which was called the cerebrum. Now, looking at this picture, you can see that it has a nice glistening to it. That's going to be the pia mater there. Also, the brain's mainly made up of fat. And if you take the human brain out of the body, it actually will collapse in on itself if you don't preserve it properly in special pickling type juices. You see lots of blood vessels going through the different grooves and feeding the brain. Now, each one of these grooves is referred to a sulci or a sulcus. The sulci is going to be plural for sulcus. Now with this, of course, we saw the different sulci and stuff. Now the huge swellings and sections are going to be referred to as gyri. And so we see that cerebrum, then we have that cerebellum right here. We have the inner part of the brain, which we cannot see, and then it comes on down through what we've referred to as the brainstem, where we'll find the medulla and the pons and things such as that. So moving on, looking at the brain, we have it divided up into different sections. So with these, you'll see how these different sulcus or sulci divide the brain into parts. So first, looking up, turn on my drawing tool, hold on. So first, looking up here, you have what we call the central sulcus. That's going to divide this frontal lobe from the parietal lobe. And so you'll see that sulcus come on down and divide that. You have what's referred to as a lateral sulcus, and that's going to divide that frontal lobe coming along the side from the temporal lobe, and even comes on as far as the parietal lobe section there. And so that's where it's going to stop. You'll also have some different landmarkings. The temporal lobe, of course, is going to be on the temporal side. You have the occipital lobe, which is going to be found towards the back of the head. And then you have what's referred to as the precentral gyrus and the postcentral gyrus. So those are going to be the large protuberances or sections. Coming on down further, in with this brainstem here, you're going to find your pons. Then you find that medulla oblongata, and then those aren't actually part of that cerebrum, along with this cerebellum, which is going to be a different part of the brain and not part of the cerebrum. Looking at this picture, we're looking down from on top of the brain, and so notice how the top part is going to the top part's going to be the anterior, bottom part's going to be the posterior. So this on the cadaver is going to be the right side of the brain. This over here is going to be the left side of the brain. Now, with this brain, if you look down on it, notice how it's divided into two different hemispheres. You're going to have the right hemisphere on this side and the left hemisphere on this side. It's going to be divided down through the center with what's called the longitudinal fissure. So that's what comes down and divides both of those hemispheres. Now, also looking at the right side, all of the vessels and the duramators have been removed. If you look on the left side, you do see the veins and the arteries that are still going to be attached. So looking at this view, this is going to be what we refer to as a mid-sagittal view. With this mid-sagittal view, you're going to see different sulcus or sulci here. And so with this, you see, let's start from the front. We have this frontal lobe, and you'll see how that frontal lobe has a pink section here. And then you'll see how it changes over into what's referred to as the cingulate gyrus, which is going to be through this section here. And then you'll see the precentral gyrus, which is going to be this large section here. That central sulcus, which cuts in through this section. You have the postcentral gyrus, which is going to be found here. The parietal lobe, which is going to be in the light purple going through here. The parieto-occipital sulcus, which is going to cut through this section here. And then you have that occipital lobe, which is found there in green. Now, you have white matter that's going to be going through the center part here, which is going to be connecting the left and right hemispheres together. This is going to be referred to as, corpus callosum. So that's going to be the corpus callosum section. So basically it's the axons. Let me see if it'll draw right. All of this is going to be the axons with that myelin sheath that's going to be around it. And so that's what's there that's helping to connect the right and left hemispheres of the brain together of that cerebrum. We also here on this picture can see part of that midbrain. We can see the pons and we can see that medulla oblongata. And then of course we see the cerebellum, which has kind of a cool look to it, a cool shape. And this is what it actually looks like inside of a cadaver or a human brain if you were to dissect it and cut into it. So with this picture, it's going to take a little portion of the brain and let you look at it closer. So these protuberances or these parts that kind of come up are all going to be referred to as the gyrus. Then you see the actual indentions that go in. That's going to be the sulcus. Also, this is going to show the gray matter or the cortex. The gray matter is going to consist of the cells that do all the thinking, all the processing, all the decision making. Underneath, you have the white matter. The white matter is going to be made up of the different axons and stuff that help connect the different cells together. So the portion of the brain that is actually fairly thin is going to be the thinking part of the brain, or that cortex part of the brain that covers the top portion. That inner white portion, that's just going to be there for the connectivity part. So there's going to be a portion of the brain that you can't quite see properly. So if you were to take two tools and pull back part of the frontal lobe And the temporal lobe right here, you can see inside, and this is referred to as the insula. And so you can see the protruding portions or the gyri of the insula. If you look here, this actually goes inside of the brain, so you can see what the inside portion of the insula looks like. And so you can see the different gyri there and sulci. This is looking at the brain from underneath, or an inferior view. Here you could see specific portions of the brain. For example, on this section here, you can see this is going to be part of the frontal. This is going to be part of the parietal that you can see. You can't see any of the occipital. You can see all this section here, which is the cerebellum. Up deep within here is going to be your midbrain. You can't really see that. Then, of course, you have your pons and you have your medulla oblongata, which you're going to find here. Also coming off, you can see specific parts like your optic nerves, which are going to attach into your eyes. So let's just say that these are your eyes here. These nerves would actually run out and attach into the eyes. And then if you notice all of these nerve endings through here, they're all going to exit off the brain. So they're all referred to as cranial nerves. And so they exit off and come out of the brain and attach to different nerves that go around the cranium. So this here's just a clay model that's showing different sections of the brain. And so from the front here, this is going to be your frontal section. Then of course you have your parietal section. It's not writing properly today. You have the occipital section here. Then you have your temporal section, which is going to be found here. And of course then your cerebellum, which is going to be found at the bottom. So going on into parts of the brain, starting with the frontal lobe, it's going to be the primary motor cortex. This is going to help control things such as your skeletal muscle. The premotor cortex of the frontal lobe is going to control learned motor skills of repetitious or pattern nature. So example, typing, playing an instrument, maybe things such as dance. This is going to be what we refer to as muscle memory. Then you have what's referred to as the Broca's area. This is going to control things such as muscles that are involved in speech. So usually it's going to be in one hemisphere of the brain. Then you have what's referred to as the frontal eye field. This is going to control voluntary movement of the eyes. So you do have skeletal muscles attached to the eye that are going to help you look around and move your eyes up and down, or even roll your eyes. Also you're going to have the prefrontal cortex. This is going to be a multimodal association area that's going to be involved in cognition, intellect, and in personality. And so looking here, you have that prefrontal cortex, you have those motor areas, so it's going to show you where all of these things are going to be found. Going into the parietal lobe, this is going to be the primary somatosensory cortex. Here is the first one. It's going to receive information from sensory receptors in the skin. So it's going to pick up on any type of information, whether it's hot, cold, temperature related, things are rough, things are painful. All that information is going to come in through that primary somatosensory cortex. Then you're going to have the somatosensory association cortex. That's going to integrate that sensory information to produce an understanding of the object being felt. So it's going to be picked up from the primary somatosensory cortex, brought in, and then that somatosensory association cortex is going to make sense of it. All right, so looking here, this is going to just show different portions of the brain and different things that they'll control or different things that they'll pick up information on. And so I'll give you a moment to look at this and see those different places. And of course, this is a sensory map that your brain makes for these things. Next, going on to the occipital lobe. The first portion of the occipital lobe we'll talk about is going to be the primary visual cortex. That's going to receive information from the retina of the eye. And so the retina of the eye is simply going to be a screen of cells that are going to pick up on information that's being focused on it. So the light is going to come in through the opening. It comes through the cornea, through the pupil, which is the opening, and it actually gets focused onto the retina using the lens. Now that lens can kind of plump up if something's close, or if you have to squint to see something far away, it might stretch itself out. So its main job is to focus whatever you're trying to look at on that screen, which is going to be found on the back part of the eye, which is the retina. And then that information, once it's picked up by those cells, is going to be sent through the neural or the optic nerve into the brain, and is going to go to this primary visual cortex. Visual association area is then going to use past visual experiences to interpret this visual stimuli that it's seeing. The next section is going to be the temporal lobe. So it's the primary auditory cortex, so it's going to interpret pitch, loudness, and location of sounds sensed by the inner ear. Then we have our auditory association area that's going to perceive the sound stimulus. So it's going to perceive it as a scream, perceive it as noise or music, or maybe just basic conversations and talking. Next in the temporal lobe, we have what's referred to as the olfactory cortex, and that allows for the awareness of smells and odors. And so looking at these different sections of the brain, you can see those here. So for example, with taste and gustatory, you can see that on that inner section there. And the olfactory, which is going to have smell. And then looking around just at the different sections that we have. So let's get this here. All right, so with the frontal lobe, it's going to coordinate information from other areas, control some behaviors. That's going to be things like your reasoning skills. So we're going to find that here. Skeletal muscle control and movement is going to be found in this orange region here. And then you have your primary cortex, which is going to be in red. You have sensory information from things such as your skin, the musculoskeletal system, the viscera, which is going to be found around the organs, and the taste buds are all going to be found in this section here. And then the section of vision is going to be back towards the back portion of the brain. So you have the visual cortex and you have the visual association area that's going to be coming in. Now with this orange section through here, that's going to be with your hearing. So it's auditory association and auditory context. Next, we have the corpus callosum. This is going to be a large band of neural fibers that we see here. It connects the two brain hemispheres together and is going to carry messages between these two hemispheres. Now, when they used to go in and do lobotomies, they would actually go in and they would sever this section. And that way, the right side couldn't contact or couldn't coordinate with the left side. So they would sever these. And when they did that, that completely changed the person's personality. So they might have been somebody who was difficult to deal with. They might have been loud and boisterous and hard to control. Usually they had some type of mental ailment. And so when they went in and did this, it made these people very docile. And so they really didn't fight back. They didn't have much of a personality after that. And in some cases, they even needed help being fed after having the lobotomy done. So this is a view of the fibers of the corpus callosum. So basically the right hemisphere and the left hemisphere, they each have sections that can do specific jobs. And so they'll divide themselves up, and one side will function working on one thing and another side will function working on another. And then these fibers will help connect them through the corpus callosum and help these two hemispheres talk to each other and help them to regulate what they're doing and let each side know what the other side's doing. And so here we have different things such as association fibers, which you'll see take signals or information from one section to the next, not too far away. Then you have what's referred to as commissural fibers. These are going to be found between the hemispheres, so you see it connecting the right hemisphere and the left hemisphere together. You see what's referred to as projection fibers, and that's going to be found in the cerebral cortex to the lower area, and that's going to help connect things such as the corona radiata and internal capsule. And so those are going to be the different fibers that you're going to see that are going to help make up that corpus callosum. And so here's looking at an actual brain and what these fibers actually look like within the brain. And you see that corpus callosum in the center, helping to connect those two hemispheres together. Here's going to be a sagittal view or a mid-sagittal view where it cuts down the center. And so you see those association fibers, you see the commissural fibers, and then you see the projection fibers. So going into the basal nuclei, it's going to be located deep within the cerebral white matter. It's actually going to be made up of a caudate nucleus, a putamen, and a globus pallidus. These are going to function to inhibit unnecessary movement, and they're also going to play a role in cognition and emotion. They're going to be important in performing specific motor functions within the body. So looking here, you see the head of the caudate nucleus, you see that putamen, and the globus pallidus. And this is going to be a transverse section or cut of an actual human brain. And this is what these sections look like. So looking at this picture here, you'll see through the center, we're looking at the actual diencephalon here. We're looking at the midbrain. We're looking at the pons and the medulla oblongata. So that's actually going to be this picture here. If you look at it, the colored in section through the center is actually this. So let's break it down again. We have that diencephalon, which is going to be made up of the thalamus and hypothalamus. That thalamus is responsible for relaying and processing sensory information. You had your hypothalamus, which is going to control emotions, autonomic functions, and your hormone production. Then we had that midbrain, pons, and we'll talk about the medulla oblongata. So let's start with that diencephalon that we were looking at. So we said it consisted of the thalamus, hypothalamus, and it actually has an epithalamus. The thalamus is going to be considered the gateway to the cerebral cortex. All the sensory information here, except smell, must pass through the thalamus to get to the cerebral cortex. And so looking here, you see where the different things are going to come in through into the diencephalon. So we can look at it from this section here. And this is going to be the main thalamic nuclei. And this is looking at it from inside of an X-ray. And we're going to find this section here. With the hypothalamus, that's going to be the center for emotional response. So that's where we're going to have responses such as pleasure, fear, rage, and sex drive. Regulation of sleep and wake cycles are going to be found in the hypothalamus. So this is going to be your biological clock or your circadian rhythm, which is going to keep you going. It also controls the endocrine system functions, and that's going to be your different hormones and things that are being secreted. And it regulates things such as hunger, thirst, and body temperature. So looking in here, we see the nuclei of the hypothalamus. And with that, you're going to find different sections. And so the hypothalamus is found all through here. And so looking at this, the first section we're going to look at it from here. That's going to be the medial, peripheric, or blood pressures controlled here. Then we're going in, and you have your supraoptic nucleus, which is found here. The anterior hypothalamic area, or body temperature, can be found in this blue section here. Water balance and stress, found in this purple section. You'll have lateral hypothalamic area, which is going to be in the green. You have dorsal hypothalamic area. You'll have the GI tract, which is going to be controlled through the center here. You also have ventromedial or satiation, when you are full, through that red section. You have shivering, which will be controlled through this orange section. And then you have feeding or food urges, which are going to be found here. And so going in and just looking at it a little bit closer. So with the paraventricular nucleus, that's going to secrete things such as oxytocin, which stimulates smooth muscle contractions in the uterus and mammary glands. So with the oxytocin, that's going to be a hormone that is secreted to actually start labor and it continues to be secreted until that baby is born. It stops producing oxytocin once the baby and the placenta has been delivered. Also, it's going to help with bonding between mother and child, and it helps with the mammary glands. So prolactin is going to make milk. Oxytocin is going to be responsible in releasing the milk from the milk ducts. Next, going into the preoptic area, that's going to help regulate body temperature by control of the autonomic centers of the medulla oblongata. And see these autonomic centers here, you have the sympathetic, which is going to be your fight or flight, and the parasympathetic, which is just your everyday taking care of your body. So remember, sympathetic, fight or flight, that's going to raise your heart rate, that's going to cause you to release adrenaline and things like that. The parasympathetic is going to be how you should be handling everyday events. So with that, it controls heart rate and blood pressure by regulation of the autonomic centers of the medulla oblongata. Next, going into the lateral tuberal nuclei, that's going to help produce inhibitory and releasing hormones that control the endocrine cells and anterior lobe of the pituitary gland. Now with that anterior lobe, that's going to be the front portion. And then, of course, you have the posterior lobe. With the anterior lobe, that's going to make most of our hormones. So if it is a releasing hormone, it's going to tell that area to release these specific hormones. If it's an inhibiting hormone, it turns those hormones off. The different ones that you're not going to find in the pituitary section, which we'll cover later on in next semester's chapter, will be things such as oxytocin and ADH, or antidiuretic hormone. Those are going to be the ones that are controlled by the posterior pituitary. Next, we have the mammillary bodies. Those mammillary bodies are going to control feeding reflexes, things such as licking and swallowing. Then we have the suprachiasmatic nucleus. That's going to regulate daily circadian rhythms. So wakes you up, helps put you to bed, and basically controls that biological clock of yours. Then we have the supraoptic nucleus that we're going to find, and that's going to be responsible for secreting antidiuretic hormone and also restricts water loss by the kidneys. With the epithalamus, it's going to be basically the pineal gland is going to extend from the posterior border of the thalamus. That pineal gland is going to produce a hormone called melatonin, which regulates sleep and wake cycles, so your circadian rhythm. So looking inside of the head, it's going to be a very, very small gland that you see back there, and that is going to be the pineal gland. It's circled in red. Going to the brainstem, it's going to consist of the midbrain, pons, and medulla oblongata. So looking at this a little closer, you can see these different parts shown here in this picture. With the midbrain, it's going to coordinate head and eye movements when we visually follow a moving object. It's also going to be responsible for a startle reflex that's going to be stimulated by sound. And so looking here at the midbrain functions that we'll see, first you have what's referred to as the roof or the tectum. You have the superior colliculi. It's going to integrate visual information with other sensory input. It initiates reflex responses to visual stimuli. Then you have what's referred to as the inferior colliculi. That's going to relay auditory information to medial geniculate nuclei, and it initiates reflex responses to auditory stimuli. Now, walls and floor. It's going to be made up of the substantia nigra. That's going to regulate activity in the basal nuclei. Then you have what's referred to as the red nucleus. That's going to help with subconscious control of the upper limb position and background muscle tone. And then the reticular formation, which is there for autonomic processing of incoming sensations and outgoing motor commands. So it can initiate involuntary motor responses to stimuli and helps maintain your conscious. So other nuclei centers, you're going to have the nuclei associated with cranial nerves 3 and 4. Down at the bottom, the white matter is going to consist of cerebral Pentacles, sorry. They're going to connect primary motor cortexes with motor neurons in the brain and spinal cord. They're going to help carry ascending sensory information to the thalamus. So this picture here is just going to show you the, hold on. Took it a second, let me exit. Let's try that again. So this section here is going to show you the different nuclei that you find throughout this section. And so these are going to be the ones we just talked about. Notice how there's a lot of this white matter here. That's going to be that matter that is going to contain lots of axons that connect cells to other cells and send a lot of information. So there's a lot of connectivity here. So going into the sections, we have first the pons. It's going to relay information between the motor cortex and the cerebellum. Remember, the cerebellum is going to be the section we found that kind of looks like cauliflower that's going to be in the posterior section of the brain. This is going to be, the pons is going to be involved in regulating the rhythm of breathing. So this is going to control your breathing, and you really won't think about it much. It's going to be kind of an involuntary type of control. And so looking here at the pons, you see with the white matter or that connective matter, you're going to have what they call descending tracts. Give me a second. All right, so you'll see these descending tracts through here and through here. They're going to carry motor commands from higher centers to nuclei of cranial or spinal nerves. Then we have what's called ascending tracts. Those you're going to see there in blue. Notice how they're coming up. Those are going to carry sensory information from the brainstem nuclei to the thalamus. Next, you'll see the transverse pontine fibers. With these fibers here, they're kind of larger parts through that section. They're going to interconnect the cerebral hemispheres together. Going down to the gray matter, remember these are going to be the cells that actually contain the neurons that are going to do the thinking. And so with the gray matter, here we have the aponostic and pneumotaxic centers. They're there to adjust activities of the respiratory rhythmicity centers in the medulla oblongata. Next, you have what's called the reticular formation. And notice this reticular formation is going to travel up and down through this section of the brain here. That's there for autonomic processing of incoming sensations and outgoing motor commands. Next, you have the nuclei of cranial nerves 5, 6, 7, and 8 in part, which are going to relay sensory information and issue somatic motor commands. Then you're going to have other nuclei and relay centers there that are going to relay sensory and motor information to the cerebellum. And so here you're just seeing another transverse cut where you're looking down. You can see how these peripheral nerves are coming in and connecting directly into this brainstem. Going on to the medulla. The medulla is going to control things such as heart rate, blood vessel diameter, respiratory rate, vomiting, and coughing. And so looking at this section of the medulla, this is going to be a transverse section that you can see, and this is going to go over the different parts here. So remember, our gray matter is going to be our section that's going to contain the cells that do the thinking or do the controlling. And so here, first, we're going to have the inferior olivary complex that's going to relay information from the red nucleus, other midbrain centers, and the cerebral cortex to the cerebellum. These different reflex centers that you're going to see, first you have your cardiovascular centers. They're going to help regulate heart rate and force of contraction. Then you have the respiratory rhythmic centers. That's going to set the basic pace of respiratory movements. Coming on down, you have the Gracile nucleus and the cuneate nucleus. These are going to relay somatic information to the thalamus. And then your other nuclei of cranial nerves, they're going to be there for sensory and motor nuclei of the 5 cranial nerves. And those nuclei will relay ascending information from the spinal cord to the higher centers. Next, you find your reticular formation, which we looked at a little bit earlier. That's going to contain nuclei and centers that regulate vital autonomic functions and extends into the pons and midbrain. Now going into that white matter, that's that connective matter, that's going to link the brain with the spinal cord. The cerebellum is going to help process information from the motor cortex, proprioceptors, visual pathways, and equilibrium pathways to provide for coordination of skeletal muscle movements. And so looking here, this is the one that we said kind of had the cool look to it, kind of looks like cauliflower. The gray matter, or the cells that are controlling the thinking and what's going on, you have first the cerebral cortex, which is going to be involuntary coordination and control of ongoing body movements. Going inside, you see that cerebellar nuclei. There, it's going to have involuntary coordination and control of ongoing body movements as well. Going over to the arbor vitae, that's going to connect the cerebellar cortex and nuclei with cerebellar peduncles. And then coming on down, you have the cerebellar peduncles, which are going to link the cerebellum with the midbrain and diencephalon and cerebrum for the superior section. That middle section is going to carry communications between the cerebellum and the pons. And then the inferior section is going to link the cerebellum with the medulla oblongata and spinal cord. So looking at that, this is what it's actually going to look like from a cadaver brain if you cut inside. Now we're going to talk about the reticular formation. So with the reticular formation, it's going to maintain the alertness in the cerebrum. It's going to act as a filter for stimuli. So actually it's going to filter out about 99% of all stimuli. It's going to be important for you to be able to pick up on just what's important to keep you going. If you were to take in all the stimuli that's around you, you would go crazy. Also, with these reticular formation, it's going to help you regulate skeletal and visceral muscle activity. So here, for example, let's take the eye. That information is going to come in. It actually gets projected onto the back of the eye, which is going to be where that retina is. It's going to come in through the optic nerve, come in to these input centers here, and then go through the brain to be processed and function. Here we see the limbic system. It's not actually going to be like an anatomical part of the brain, it's more of a functional part of the brain. So you'll see here part of the amygdala, you see the hippocampus, you see the thalamus and the hypothalamus. And so when you put these together, this limbic system basically is going to be what we refer to as our emotional brain. So it puts together our emotions. So looking here, we see the different sections. And so with the diencephalonic components, we have the thalamus and hypothalamus. With the other components, it shows that reticular formation. Coming over to the limbic lobe of the cortical areas, you have what's considered to be the cingulate gyrus. You have the parahippocampal gyrus. And then you have the dentate gyrus. And then you come down, you have the tracts and the amygdaloid body and the hippocampus. And so here is somebody, they are doing a dissection of the brain and the central nervous system. Let's see if this will work for you.
The adult human brain weighs approximately 1.5 kilograms, making up less than 2% of a person's body weight. And yet, it defines our humanity and makes us the individuals that we are. The brain is responsible for the generation of language and thought, attention, consciousness, memory, and imagination. In order to fit into the skull and accommodate the massive number of neurons and connections needed, the brain is highly folded. This results in the creation of gyri or ridges and sulci or furrows. If we were to unfold the entire human brain, it would take up approximately 1 square meter. Perhaps the most impressive feature of the brain is the amount of connections formed between neurons. There are an estimated 86 billion neurons in the brain, each of which forms an average of 7,000 connections with other neurons, resulting in between 100 and 500 trillion synapses within the brain. In an attempt to conceive of the enormity of this system, the number of neurons in the human brain has been equated to the number of stars in the Milky Way.
The adult human brain weighs
Now that we have
approximately
begun
1.5
to appreciate the
kilograms,
complexity
making
of
up
the
less
human
than
brain,
2% of a
let's
person's
begin
body
to
weight.
examine its structure.
And yet, it defines our humanity and makes us the individuals that we are. The brain is responsible for the generation of language and thought, attention, consciousness, memory, and imagination. In order to fit into the skull
brain
and
can be divided
accommodate the
into
massive
functional
number of
and
neurons and
anatomical
connections needed, the brain
We
is
will now
highly
start
folded.
with an
This
anatomical
results in the
overview
creation
to
of
establish
gyri
a
or
common
ridges and
terminology
sulci or
and to
furrows.
describe the
If we were
areas
to
of the
unfold
brain.
the entire
The
human
brain has
brain, it
multiple
would take up
surfaces.
approximately
Here
1
we
square
have
meter.
the superior surface.
Perhaps the
and
most
this
impressive
here is
feature
the
of the
inferior
brain
surface.
is the
There
amount
are
of
also
connections formed
anterior
between
and
neurons.
posterior
There are an
aspects to the
estimated
brain.
86 billion neurons in the brain, each of which forms
Here's
an
a
average
section
of
through the brain
7,000
within the
connections
skull,
with other
and we
neurons,
can appreciate
resulting
the
in
axes
between
within
100
the
and
CNS.
500
Connections
trillion
can travel
synapses
either
within the brain.
towards the anterior
In an
pole,
attempt to
here,
conceive of the enormity of this system,
or
the
towards
number of
the
neurons in the
inferior end
human
of the
brain
spinal
has
cord.
been equated to the number of stars in the
Fibers
Milky
can
Way.
move either rostrally
Now that we have begun
towards
to
the rostral
appreciate the
pole
complexity of the
or
human brain,
caudally
let's
towards the
begin
caudal
to examine
pole.
its structure.
Now, let's look at these component parts of the brain. This is the forebrain, which is composed of the cerebral hemispheres and deep structures. The right and left hemispheres are roughly symmetrical.
The brain can be
and are
divided into
connected to a
functional
corpus
and
callosum,
anatomical
which
regions.
You can see here.
We will now start with an anatomical
Here, we
overview
have
to
cut the brain
establish
in
a
half,
common terminology
separating the two
and to
hemispheres
describe
from
the
each other.
areas of the brain. The brain
This
has multiple
is the cut
surfaces.
surface
Here
of
we
corpus
have
callosum,
the superior
the
surface,
largest white matter
and this
tract in the
here
brain.
is the inferior surface.
Impressively, it
There are
consists
also
of an
anterior
estimated
and
200 to
posterior aspects
250
to the brain.
million projections. This structure here is the
Here's
thalamus.
a section through the brain
And together
within the
with the
skull,
hypothalamus
and we can
here,
appreciate the axes within the
and the
CNS.
subthalamus, which you
Connections
cannot see,
can travel
it makes
either
up the
towards the
diencephalon.
anterior pole, here,
The cerebral hemispheres and the
or towards the
diencephalon
inferior
together
end of the
comprise
spinal
the
cord.
forebrain. We now
Fibers
move into the
can move
brainstem,
either
which is
rostrally
caudal to the
towards the
diencephalon.
rostral pole
The brainstem can
or
be divided
caudally
into three
towards
parts.
the caudal pole.
The midbrain,
Now, let's
located
look at
here,
these component
just
parts
caudal
of the
to
brain.
the thalamus, contains the large fiber bundles, the cerebral
This is the forebrain,
peduncles,
which is composed of the cerebral
that connects the
hemispheres and
forebrain
deep
with
structures.
all caudal
The
structures.
right and left hemispheres
The
are
pons
roughly
is
symmetrical
located caudal
and
to
are
the
connected
midbrain.
through corpus
It is
callosum,
connected
which
to
you
the
can see
cerebellum
here.
through the cerebellar peduncles.
Here, we have cut the brain
The
in
most
half,
caudal part of the
separating the two
brainstem
hemispheres from
is
each
the
other.
medulla. It is continuous
This
with the
is the
spinal
cut
cord as it
surface
exits
of corpus
the foramen
callosum,
magnum of the
the
skull.
largest white matter
The
tract in the
cerebellum
brain.
is embryologically
Impressively,
part
it consists
of the pons,
of an
but
estimated
its functions
200
are
to
so
250
distinct that it is
million
now
projections.
considered its own entity,
This structure here
separate from
is
the
the
brainstem.
thalamus.
The
And
cerebral
together with the
hemispheres can be
hypothalamus
divided into
here
lobes along some key
and the
surface
subthalamus,
landmarks.
which you cannot see,
The
it makes
central
up the
sulcus
diencephalon.
separates the
The
frontal
cerebral
lobe
hemispheres
from the
and
parietal
the
lobe.
diencephalon together
The lateral
comprise
fissure
the forebrain. We
separates
now
the
move into the
temporal lobe
brainstem, which
from
is
both the
caudal
frontal
to the
and
diencephalon.
parietal lobes.
The brainstem
The
can be
occipital
divided into
lobe
three
is
parts.
separated from the
The
parietal
midbrain,
lobe via
located
the
here just
parietal
caudal to the
occipital
thalamus,
sulcus,
contains the large
which you
fiber
can see on
bundles,
this
the
medial
cerebral
aspect of the brain.
peduncles,
You can draw
that
a
connect
line
the
onto the
forebrain
lateral
with all
surface
caudal
right
structures.
here
The
to
pons is
differentiate
located
between the
caudal to the
occipital
midbrain.
lobe and
It
the
is
parietal
connected to the
and temporal
cerebellum
lobes.
through the cerebellar
In this
peduncles.
medial view of the brain,
The
you
most
can
caudal
identify
part of the
a
brainstem
continuous
is
strip
the
of
medulla.
cortex,
It is
which swings
continuous
around
with the
surface
spinal
of the
cord
brain.
as it exits
This
through
lobe has been
foramen
dubbed
magnum
the
of the
limbic
skull.
lobe due to its
The
intimate
cerebellum
relationship
is
with the limbic
embryologically
system.
part of the
It
pons,
spans
but
the
its functions
frontal,
are so distinct that
parietal,
it is now considered
and
its
temporal
own
lobes.
entity, separate from the
Deep within the
brainstem.
brain are spaces
The cerebral
filled with
hemispheres can be
cerebrospinal
divided
fluid,
into
or
lobes
CSF.
along some key surface
This is a 3D
landmarks.
reconstruction of the
The
ventricular
central
system.
sulcus
You can see the two
separates the
C-shaped
frontal
lateral
lobe
ventricles
from
in
the
the cerebral
parietal lobe.
hemispheres. They
The
have
lateral
an anterior
fissure
horn deep within the
separates the
frontal and
temporal
parietal
lobe
lobes of the forebrain,
from both the
a
frontal
posterior
and
horn which
parietal
extends
lobes.
into the
The
occipital
occipital
lobe,
lobe
and an
is
inferior
separated
horn
from the
which extends
parietal lobe
into the temporal
via
lobe.
the parietal
These lateral
occipital
ventricles are
sulcus,
connected to the third
which you
ventricle
can see
in the
on this
midline.
medial
The
aspect
third
of the brain.
ventricle is connected
You
to the
can
fourth
draw a
ventricle
line
through the
onto the
cerebral
lateral
aqueduct.
surface right
On this
here
mid-sagittal
to
section of the brain, we
differentiate
can
between
identify
the
different
occipital
components of
lobe
the ventricular
and the
system.
parietal and
This
temporal
here is the
lobes.
lateral ventricle.
In
The
this
anterior
medial view
horn
of
will
the brain,
extend
you
anteriorly
can
here into
identify
the
a
frontal lobe,
continuous
and the
strip of
posterior horn will
cortex
extend
which
into the
swings
occipital
around
lobe.
the surface of the
This
brain.
here is
This
the
lobe
third
has been
ventricle.
dubbed the limbic lobe
The
due
thalamus
to its
is on either
intimate
side of the
relationship
third
with the limbic
ventricle.
system.
The third
It
ventricle
spans
connects
the frontal,
to the fourth ventricle via
parietal,
the cerebral
and
aqueduct
temporal
right
lobes.
here. This
Deep
here is
within
the
the
fourth
brain are
ventricle
spaces filled
at
with
the level of the
cerebrospinal
pons
fluid,
and
or
medulla.
CSF.
The fourth
This
ventricle
is
is
a
going to
3D
close off into
reconstruction
the
of the
central
ventricular
canal.
system. You can
Let's
see
look at
the
the
two
various planes the
C-shaped
brain
lateral
can be sectioned
ventricles
in.
in the cerebral
This is a
hemispheres.
very important
They have
concept
an
because
anterior
imaging of the
horn
brain uses
deep
these
within the
planes.
frontal and
I'm going
parietal
to cut
lobes of
this
the
brain in
forebrain,
the
a
coronal plane.
posterior horn which extends into the occipital lobe, and an inferior horn which extends into the temporal lobe. These lateral ventricles are connected to the third ventricle in the midline. The third ventricle is connected to the fourth ventricle through the cerebral aqueduct. On this
So you can
mid-sagittal
now see
section of the
corpus
brain, we
callosum
can
here, and
identify
we've
different
started to look
components of
into
the ventricular system.
So this is the
This
anterior
here is
tip
the lateral
of the
ventricle.
anterior horn here.
The anterior horn will extend anteriorly here into
So
the
this
frontal
next
lobe,
slice will take
and
us
the
into
posterior
the
horn
anterior
will extend
wall of
into
the
the
temporal
occipital
lobe
lobe.
right here.
This here
Those
is the
pieces
third
might fall
ventricle.
off because they're not
The
connected
thalamus
to
is
the
on
plane
either
that I'm
side
cutting
of the
in.
third ventricle. The third ventricle
go.
connects
Let's have a look
to
at
the
that.
fourth ventricle via the cerebral
All
aqueduct.
right, so what you can
right
see on
here.
this slide is really the
This here is the fourth
lateral
ventricle
again.
at the
That's
level
the
of the
head
pons
of the
and
caudate
medulla.
nucleus right there.
The fourth ventricle is going to
And the
close off
putamen.
into the central
Here,
canal.
this is the anterior limb of the
Let's
internal
look at the
capsule.
various planes a brain can be sectioned in. This is a very important concept because imaging of the brain uses these planes. I'm going to cut this brain in the coronal plane.
All right, so in this section now, you can see the two lateral ventricles, as well as the third ventricle here on either side of the thalamus.
So you can now see corpus callosum here, and we've started to look into the ventricular system. So this is the
All
anterior
right, so
tip
here
of the
in this
anterior
section now,
horn.
you can again see the two lateral ventricles. Here we're going
So
into
this
the
next
cerebral
slice will
aqueduct
take us
on our
into
way
the
to the fourth
anterior
ventricle.
wall of the temporal lobe
This
right here.
is
And those
the
pieces
inferior
might
horn of the
fall
lateral
off because
ventricle.
they're not connected to the plane that I'm cutting in. There we go. So let's have a look at that. All right, so what you can see on this slide is really the lateral ventricle again. We've got the head of the caudate nucleus right there, and the putamen.
So
here,
in
this is the
section
anterior
here
limb
again,
of the
here
internal
are
capsule.
the two lateral ventricles. Here again is the inferior horn, the hippocampus, right there in the floor of the inferior horn. All of this here is the thalamus. We're quite posterior now.
All right, so in this section now, you can see the two lateral ventricles, as well as the third ventricle here on either side of the thalamus.
Right here, we are in the midbrain and the pons. So it's a bit of an oblique section here through the brainstem, but you can see the cerebral aqueduct here, and of course this is the pons.
All right, so here in this section now, you can again see the two lateral ventricles. Here we're going into the cerebral aqueduct on our way to the fourth ventricle. And this here is the inferior horn of the lateral ventricle.
You can see that we're now getting into the posterior horn of the lateral ventricle. This is the very, very tail end of corpus callosum there. You can see the cut surface here of the cerebellum. It's the middle cerebellar peduncle moving in there. And here you can see the central canal. It's going to open up into the fourth ventricle, which will lie
section here again, here are the two lateral ventricles. Here again is the inferior horn, the hippocampus, right there in the floor of the inferior horn. All of this here is the thalamus. We're quite posterior now, as you can tell in the brain. So all of this is thalamus here. We're actually getting into the posterior parts of the thalamus there.
Gray and white matter.
Right here, we
White
are
matter
in the
is the
midbrain
sum of all
and the
myelinated
pons. So it's a
axons
bit
and
of an
tracts
oblique
as
section
they
here for the
travel through the
brainstem. You
CNS.
can see the cerebral aqueduct
Gray
here.
matter
And
is
of
the
course,
sum
this
of all nerve
is the
cell
pons.
bodies. It can be seen here along this cortical band on the surface of the brain, which is why an increased surface area is so important to accommodate the large number of neurons in the cortex. Gray matter
Alright, so
can also
in this
be found
section here,
in these deep
you can see
nuclei
that we're
of the
now getting
forebrain,
into the posterior horn of the
including
lateral
basal
ventricle.
ganglia.
This is the very,
limbic
very tail
structures.
end of corpus callosum there. You can see the cut surface
We're going to
here
cut this
of the
brain in a
cerebellum.
horizontal
It's the middle
orientation.
cerebellar peduncle moving in there. And here you can see the central canal. It's going to open up into the fourth ventricle, which will lie just on the other side of that opening there. In this coronal section, we can see both gray and white matter. White matter is the sum of all myelinated axons or tracts as they travel through the CNS. Gray matter is the sum of all nerve cell bodies.
Okay, we're now
It
going to
can be
cut through
seen
the
here
brainstem
along
here
this cortical band on the
in an
surface of the brain,
axial orientation.
which is why an increased surface area
So here's
is
the
so
midbrain.
important to accommodate the large number of neurons in the cortex. Gray matter can also
And here
be
we've
found
cut
in
through the
these deep
pons in the
nuclei
superior
of the
cerebellar
forebrain,
peduncle as it projects to the
including
cerebellum.
basal ganglia and limbic structures.
So this is the last
We're going to
section
cut
here
this
through the
brain
pons.
in a
You can see
horizontal
some of the deep
orientation.
cerebellar nuclei here. In this cut here, you can see the inferior part of the cerebellum and then part of the open medulla. And here you can see the closed medulla as the ventricle has closed over the central canal here. Finally, the brain can be cut along the sagittal plane. Here we have cut the brain along the mid-sagittal plane to separate the two hemispheres.
Okay, we're now going to cut through the brainstem here in an axial orientation.
With
So
this
here's the
overview,
midbrain.
we now have a common terminology as we begin our journey through the brain.
And here we've cut through the pons in the superior cerebellar peduncle as it projects to the cerebellum.
So that is going to end us
So this
with
is the
our
last section here through
on
the pons.
the
You can
brain
see some of
and
the
on
deep
the
cerebellar
cranial system.
nuclei here.
I'll see you next semester, and we will pick up with our actual body systems
In
from
this
there.
cut here, you can see the inferior